Hello, my name is Pratyush Chira, and today I'll be presenting my journal discussion on the virtual core flow cytometry paper. So, some background on what flow cytometry is. At its core, flow cytometry is a technique to measure the physical and chemical properties of a sample through the use of a laser emitter. This is done by floating the sample, usually a type of cell, through a fluid, and then passing that fluid through a nozzle and exposing the cells one by one to the laser emitter. From here, the refracted light is passed through a series of prisms and mirrors and collected by a series of detectors. These signals are then processed by a computer system in order to create a system of wavelengths that can then be used to categorize and evaluate the sample. However, one common limitation of this method is the need to ensure that the cells maintain a straight line and are equally exposed to the laser emitter, as appearing outside of the focal point of the laser would result in errant signals. To achieve this, there are several standard methods. The most basic is to float the cell sample in a sheathing fluid that prevents it from moving too far. Another method utilizes electrical fields in order to move the cell samples and ensure that they are maintained at the center of the flow. Another method utilizes machine channels. These are, of course, microchannels to guide the cells past the laser in a single file line. And the final method utilizes an auditory transducer in order to shift the cells into a straight line. However, a limitation of all of these methods is the need of a sheathing fluid to ensure the flow. Now, the problem with the use of a sheathing fluid is that the fluid itself is relatively expensive, particularly for the amount needed to perform the process. A large amount is needed for a relatively small amount of samples, and the fluid is almost impossible to recycle without expensive equipment, and thus is usually not worth doing. This, in turn, limits the availability of this technique to larger, more well-established labs, and also limits the use of this technique outside of a clean laboratory setting, as you need to ensure that the sheathing fluid is constantly flowing and constant maintenance is performed on the pumps to ensure that the machine doesn't get damaged. All of this increases the cost of experiments and limits the technology to only a lab setting and does not allow for practical field use. On the right, we can see an example of one such device, a simple flow cytometer, and you can see that a large percentage of the image is taken up just by the two sheathing fluids needed for the system to operate. In order to work around this limitation, this paper proposes the idea of a virtual core flow cytometry system. At its core, this method attempts to reduce fluid usage by creating a virtual region of interest outside of which samples will be ignored, and the refracted light from such samples will basically not be considered for the final computation. In order to achieve this, a system of light emitters, magnifiers, relay lenses, as well as a series of complicated detectors are utilized to create a virtual region that is extremely small but is within the focal point of the laser emitter. And this allows for the filtering to ensure that only the cells that appeared within the focal point are considered for further computation. In order to evaluate the results of this technique, the lab performed two experiments. First, they attempted to separate between cells with varying CD4, CD8 expression. This is an important hurdle to surpass, as it is a very common experiment for the majority of cancer cell studies, particularly those attempting new treatment methods. As such, it is a very common use case for flow cytometers. Now, on the right, you can see the result of a standard flow cytometer with a sample of various T cells, and you can see that it pretty distinctly forms three clusters. When we remove the sheathing fluid, we can see that the resulting signal is an incredibly blurred image, as false positives basically affect and cause artificial diffraction that is detected as cells of various scores, even though they really don't exist. Once we add the flow cytometer limitation of only being considered within that focal point region, we see that there are far fewer false signals, and the three clusters once again reemerge. However, we should be noted that the total number of signals has been reduced, as they are not collected, they are simply rejected. However, it should also be noted that the lab was able to demonstrate that this did not result in any false signaling and was within 2% of the standard method. A further experiment that the lab utilized was to run a sample of seawater through both their system and through the flow cytometer. Here you can see the result of their system, and you can see 4 tight clusters forming. Once we look at the standard system, we can see that these same 4 clusters appear, though they are of far greater magnitude. The results of this experiment show that this method has a similar standard when it comes to the accuracy of the clusters, though it has lower intensity and individual cell counts within each of those clusters. However, it uses significantly less fluid and also allows for the lab to reanalyze samples as they are no longer being diluted by the sheathing fluid, and thus you can rerun the same sample over and over again until the sample itself is degraded by repeated exposure to light. This system allows for the reduction in size of flow cytometers and thus allows for the field use of these devices. One proposed method and location is within the bulkhead of ships to detect the materials found within water in the ballast tanks. A future development of this project that was considered by the authors was to increase the complexity and structure of the detectors to better ignore the false signals while more accurately resolving the true focal signals.